To first begin with, I would like to introduce to you our main title of the talk, which is Uncovering Cellular Heterogeneity with Multi-Omics Approaches for Single-Cell Analysis. And to get started, I would actually first begin by explaining what is Singleron doing in the space and what is single-cell sequencing, as I got to know from the poll earlier organized by Elena, but a lot of people are still interested in getting to understand what's the benefit of using single-cell sequencing. So that's one of the sessions that I will have. And by the end, we will discover about our technology and the multi-omic solutions that we offer. So on the next slide, what I have here is our different locations. So the one location which I think would be very relevant for you would be our Michigan site and our Connecticut site, as they both are based in US. And from those sites, we cover our North American market. And we do our manufacturing in US, and we have lab support as well as technical support available in Connecticut and Michigan, both the places. We also have our headquarters in Cologne, Germany, which is our headquarters for all across the world. So that's where most of the people are situated in, but we also have offices and are growing our team in US. We also have our team in China and Singapore to cater to people across the world. And due to that, we have more than 500 plus scientific publications until now. And we are working with 6,000 plus customer organizations worldwide. Now, moving on to the next slide, I would like to talk about how single-cell sequencing and bulk sequencing can be different and what is the benefit of working with single-cell sequencing. To get started with that, I would first like to introduce bulk sequencing. So to look at bulk sequencing, it's almost like a fruit smoothie. So you don't actually get the taste of one individual fruit. You have multiple different fruits which are mixed together and you get like a pool taste. So it's an average of your entire sample. So if there's a increase in gene expression of cell type 1 and decrease in different cell type, you will get an average of that sample, which might not be the case for all the samples. You might see an increase in certain cell type in a certain gene expression while decrease in another. So to look into that, single cell sequencing can be very helpful because in this case, we are not looking at a fruit smoothie, but we are looking at a fruit basket. So you can count the different fruits, you can detect the exact flavor or the taste of each cell type, or if you would like to call it profile of each cell type, and get to know about the gene expression, and different cell types can be discovered which you might not know earlier or might have missed in the bulk seq, which can be discovered further with the single cell sequencing. So that's the benefit of working with single cell sequencing. And to talk about more about single cell seq, we actually were happy to announce that Science actually called it Breakthrough Technology of the Year for 2018, and Nature Methods called it the Method of the Year for 2019 for single cell multiomics. And that is something that we also provide, and our customers are living proof of what we actually provide. And these are a couple of our testimonials. And they have been using us for getting single cell suspension from tissues and then doing data analysis as well as library prep with our kits. So here we have customers from US, Singapore, Germany, Norway, and Spain. But we can also soon have something from Canada too onto the list, which we'd be happy to share. Yeah, so to get started with our solutions, I would like to talk about our product portfolio. And to talk about our product portfolio, I don't want to begin just from single cell sequencing. We actually are a one-stop shop beginning from tissue dissociation. So as soon as you get a tissue, we actually have a pipeline ready to help you get the ready data for your publication. And we also offer automation to help you automate the processes and not only provide like manual workflows. So you have manual workflows along with automation. And to get started, as we are talking here from tissue dissociation, which could be considered the first step, so to cater to that, we actually have a complete pipeline. So as soon as you get the tissue, you can put it in our tissue preservation buffer, which can store the live tissue without any fixatives for up to 72 hours. And in this tissue preservation buffer, you can add your sample and then store it. For example, if you receive your sample on a weekday afternoon, you can store it and would like to work it on next day morning. You can just put it in the preservation buffer, the live tissue. And then next day morning, you can take it out and use our tissue dissociation solution kit along with the units, which offer you enzymatic dissociation along with mechanical. As you can see, the teeth of the dissociation unit in a way like these, they mince the tissue, and this is happening in our tissue dissociation instrument. In this case, we have a smaller version on the screen, which is Python Junior. If you want a larger one, we also have a larger one for labs which do more of tissue dissociation. Here you can see both the versions on the screen. The left version is our 8-channel version, and the right version is our junior version. So depending on the user case, you can use our larger or smaller version. Here we have some performance data. So we have the viability and yield. You can see that the mouse tissue examples that are listed here are heart, liver, spleen, lung, kidney, and testicles. And we see more than 85% viability for most of the tissue types. So it can range from 96% to 85%, 84.94% as viability, depending on the tissue that we are looking at. And the cell yield per milligram is also depending on the tissue that we are working with. But we have experience with more than 400 plus tissue types, so this can be quite relevant. And here we have our customer testimonial with their data that they generated working on different tumor types, and then the percentage of increase in viability and cell yield in this case, depending on competition that they did a comparison with on their end. So as soon as you get like your single cell suspension from the tissue and a good cell viability and cell yield, then we can move to the next step, which is doing the single cell partitioning and lysis and barcoding. To do that, we have our scope chip, which is our main proprietary technology. You don't need any instrument to work with this. You just need a P200 pipette and you're ready to go. As you can see on the slide here, the scope chip has an inlet. I hope you can see my pointer. So you have an inlet where your pipette tip can mount, and then you have an outlet reservoir. So you add your single cell suspension into the inlet, and if you zoom into the chip by putting under a microscope, you will see multiple wells in here. And in each well, basically, you will have one cell that falls into it. And then once you have the cells that are added into the chip, then we add our barcode beads. These barcode beads are included in our kit, as you can see on the slide. And then they're added onto the chip similarly as you add the single cell suspension. And once you add the as you add the poly-T barcode beads, they capture the poly-A tail of the mRNA, and the capture is happening on the chip. And to visualize it, we can see that in the slide. You see the empty wells on the left-hand side, and then you add the cells. You will be able to see the cells. Depending on the size of the size of the cells would vary. So in certain cases, you would see larger cells, as you can see here, but in certain cases, the size would be smaller if your cell type is smaller. And then you add the barcode beads. You would see that each barcode bead goes into one well and the cells. So that's how the capture is happening. Then we add the lysis solution. And once we add the lysis solution, then the cells are lysed and the mRNA is captured by the poly-T tail of the barcode bead and it binds to the poly-A tail of the mRNA. And once that's done, then we can actually capture all the barcoded mRNA and retrieve it into an Eppendorf, as all the mRNA is labeled at this stage. To do so, we actually add a magnet on top of this chip. As you can see on the slide on the right-hand corner, we basically just have a magnet which is included in the kit. You place it on top of the scope chip and then you retrieve the mRNA. Once you retrieve the mRNA, the next step would be to go for RT-PCR and library construction. So RT-PCR will help you after this point in converting the mRNA into cDNA. And once you convert it into cDNA, you can then have a library constructed for sequencing. You can use Illumina, Element Bio, or any of the standard sequencers that are provided. And we have the reagents included until library construction into into our kits. And the benefit of using our scope chip is that you have instrument-free processing in case you are just starting with single-cell sequencing. And if you want to scale up over a period of time, you have the option to automate, which I will shortly introduce to you. And the entire workflow can be done in one day from sample to library construction, and then you can send it for sequencing. Our system is quite robust, so in case you're working with slightly dirtier sample, as you are relying on gravity for the cells to fall into the wells, the debris will float, and then in our washing steps, they can be removed. So it's quite robust as a system, and it can also help you in capturing all the gentle cell types like neutrophils, which are usually lost with other technologies, which actually rely on a system which can be harsher on the cell type. We rely on gravity, so it's quite useful for capturing fragile cells. And you can capture up to 30,000 cells per sample. So with one chip, you can do 30,000. And this is without multiplexing. We do have the option to multiplex in case someone is requiring it. But even if you don't, you can do 30,000 cells per sample. And the best part is you can do real-time quality control by just putting the chip under a microscope and visualizing it. So that's another reason that the scope chip can be relevant. And in case you want the automation, here is just a short overview about the instrument. So basically, it can do the processes that I just mentioned to you with the pipette where you do it manually. You can do it with the instrument. You load the chip, and then the chip actually is put in the instrument, and the instrument does the chip priming, cell loading, barcode beads are being loaded on the chip, cell lysis as well as mRNA capture and barcoding is done. And once it's done, then you can go for RT-PCR and library construction and so forth. So Neo can really make your life easier, and we also offer it as a demo instrument for people who would like to test it. So that's a possibility, and you can get in touch with us to know more about it. Just to give you a bit more information about the instrument, the entire runtime for it would be 38 minutes, and that will be helpful for you to run 4 samples in parallel, and you don't need to run 4 samples if you have only 2 samples, for example. So you can actually optimize it. So if you have 1 sample at a time, you can run 1 chip at a time. We do know that from alternate technologies that that's sometimes not a possibility. So this can be quite useful if you are looking into more flexible approach for your single cell sequencing. Now, I would like to shortly mention about the performance data for the instrument. So here we have 4 replicates for bone marrow in one run, and we see that different cell types are captured very well, and the population is reproducible over the 4 samples that were run in parallel, which shows how consistent the results would be with the instrument, as well as if you do it manually, it would be consistent, but just to give you performance data of our instrument. And to talk about the number of genes and cells that can be captured with our instrument, we have here human and mouse cell line that were mixed 50-50. And then we looked at the number of cells that were captured. We captured about 11,000 cells here, and our doublet rate was about 2.7%, so which is pretty low considering the number of cells that we captured. And we look at the median genes per cell, which was about 3,366 genes. and the saturation rate was 38%, so it's pretty low still. We also have the knee plot. So all this information will be available with our Cellular software, which is freely available on GitHub, and I'll mention it shortly in the coming slide too. So in case that would be of interest to you, you can actually, as soon as you get the raw data, you can download the Cellular software and do your FASTQ QC, map to the reference genome, and get unsupervised cell clustering as well as gene count matrix. And then later for secondary analysis, we have a paid software that can be used for secondary analysis and cell type annotation, can study gene expression, regulatory networks, differential gene expression, UMAP visualization. So you can get publication ready data and you don't need to code, which is a lifesaver for a lot of biology researchers who don't have a background in bioinformatics, I feel. So this is another product which I think can be useful. Moving on, I think what we have discussed until now has been our core technology, which is for single cell sequencing, which can cover single cell, single nuclei, RNA library kits, and we also have a yeast library kit if someone is studying yeast and would like to learn more. But in case you are looking to add more data along with the whole transcriptome, so an additional layer of information like multi-omics, so we have full-length mRNA kit, we have immune profiling full-length of TCR, BCR kit, and we also have targeted variant detection, time-resolved transcriptomics, glycosylation discovery kits, which can be of interest. I'll mention them shortly in the coming slides and then take my time to talk in detail about time-resolved transcriptomics, which is also quite unique to Singleron. And in case you have any questions, you can write in the chat. We'll have a session later, but I'll just quickly go through our multi-omics kits. So here we have a focus scope to begin with. Basically, you will capture the poly-T-based poly-A tail capture of mRNA, as you can see on the top. So here is my laser pointer. So you have the poly-T based capture. And then we have probes designed for up to 10 custom regions. It can be custom, or if you like, couple of our pre-validated panels. So we have blood cancer panel, lung cancer panel, clonal hematopoiesis panel, as well as EBV virus panel. And we recently launched intracellular bacteria. So if you have human cell with intracellular bacteria, you can also capture both of them parallelly, which can be quite interesting for researchers. So we have these panels available. If your gene of interest are covered, you can already order the pre-validated panels, but we can have a custom panel built for you to capture your fusion genes or viral genes or regions of interest. We also have Chromoscope, which can do transcriptome plus cell surface glycosylation. And you might be wondering how it works. So in this case, we basically have a fucose residue with a PCR handle and a poly-A tail. So this Chromoscope tag goes and binds covalently to the LACNAC region of cell surface proteins. So all the cell surface proteins with the LACNAC region would actually have this tag attached to them. So you can quantify later on by doing the bioinformatic analysis by looking at the library constructed for the cDNA of chromoscope tag and cDNA of the whole transcriptome, and you can get an idea about the quantification of glycosylation for different cell types. And this is also something which can be quite interesting for studying cancer or any disease type compared to control conditions and looking at the glycosylation patterns. Another interesting kit that we have is Mobioscope, which is full-length single-cell RNA sequencing with short-read sequencing. In this case, we basically have a technology to look into 3' or 5' based library to avoid minimum and make minimal 3' or 5' bias. And you get long-read sequencing data, like you have the insights which you might normally get from long read sequencing at short read pricing, because you would be using short read sequencers to analyze. And it can be quite useful to study splicing junctions as well as non-model organisms. How it works is that we actually capture based on the bead on the barcode bead. But once we have the C prime in library, we have another part which actually undergoes circularization. So once it undergoes circularization, it brings the 5 prime end close to the barcode. And as we have the 5 prime end closer to the barcode, we can later do reverse PCR or inverse PCR. And with this, basically, we have complementary cDNA. And in this sequence, as the barcode linker region is closer to the 5 prime end, we can have the cDNA fragments and 5 prime end library construction. So you can basically get a 3 prime end library and a 5 prime end library, and you cover the full length of the transcriptome with it. And there's minimal 3 prime and 5 prime bias as shown in the figure below. And then I would like to now talk about DynaSCOPE, which is the technology that we would like to discuss in further detail today. It helps you and enables you to detect newly synthesized mRNA at single cell level. So in this case, basically, we have a label that is added to the cells. It can be done in vitro or in vivo experiment. So you are not bound to using just in vivo models. You can also inject it into the mice or rat. And how it works is basically you have conversion of S4U label in the nascent mRNA into cytosine, while the old mRNA will have uracil, which is incorporated into the sequence after PCR as thymine. So it's converted into thymine, while the S4U incorporated sequences are converted into a cytosine analog. So this is something that I feel can be quite relevant for people who would be interested in looking at changes over a period of time. So you can see if you are working with certain drugs or you're studying developmental biology, you can have the drug inserted at different time points and have the data comparing it. So as I mentioned about the technology and how it works, I would also like to mention here that we have a bioinformatic pipeline for it to help you in this process. And here's the entire workflow. So we begin with the in vitro and in vivo labeling, and then we do the tissue dissociation, single cell suspension, cell partitioning, lysis and barcoding, as well as library prep and sequencing and data analysis. To go further into detail about it, so we have lung tissue that we actually look into the data for it. And we have new mRNA versus old mRNA, and we look at the changes over a period of time, in this case for DynaSCOPE. And we see that monocytes, B cells, and T regulator cells are transcriptionally more active in the lung tissue. And hence, we see that there is a higher change in the B cells here and the monocytes. So you see more mRNA being transcribed at the nascent stage too, compared to, for example, here we look at the fibroblast and the ECs. You don't see much of the activity in the new RNA compared to old. So this is something that can be studied using the DynaSCOPE kit. We also have some further data analysis, so you can go deeper into the individual genes and transcripts with the use of DynaSCOPE. So here we actually look into IL7R, ERHEAP45, and CLEC7A. In these cases, we see that the freshly synthesized nascent mRNA is quite stable across the old mRNA versus the new. So the new one is marked with the darker color, like the reddish, and the gray is the old RNA. While if we look at the FTPA1, LAMP3, and FTPB, we look at the freshly synthesized in B cells only. So you don't see that change over time in nascent mRNA, in endothelial cells, and macrophages. So that's only detected in B cells. While certain transcripts are much more stable across like in B cells, endothelial, and macrophages here. So you can go deeper into the data and look at it for different cell types as well as different gene expression over a period of time in nascent and old mRNA. And we also have some publications already published. I can talk about them in the coming slide. So here we have two publications which have been published using our DynaSCOPE kit, and I will go deeper into detail about the second publication in the coming slides. So here we go. We are talking about peripheral T cell lymphoma. And in this publication, which is published in Advanced Science, they are looking into T cell cancer type with very poor prognosis, and they were looking into new therapy options. And one of the ways to look into it, they targeted was the CGAS STING pathway and how it can be useful in and be used as a target for future treatments. So to do so, they actually looked at different inhibitors for CGAS and STING, as well as for TBK1 and STAR3 inhibitor. So they studied the inhibitor and looked at the cell viability over a period of time. Here you can see 24 hours and 48 hours data. And you can see that over a period of time, the STAT3 and CJAS, so the case A and case D, in both the cases, we do see that there's a significant reduction in cell viability over a period of time. And these are the cases where we do see that if we can combine CGAS inhibitor with chemotherapy, it can maybe improve in tumor cell death with CLK1 as a potential marker for treatment sensitivity. So maybe this is one of the ways that can be studied. And another thing we saw was the G150, which was studied as the marker in this case. When they looked at the flow cytometry results, we do see that the live cells portion is higher here, which is the DMSO as expected. But with G150, the CGAS inhibitor, we see higher number of cell death in the Q2 here. So this is something that can also be studied further. So like if you want to look at it at single cell level, so you can study it using DynaSCOPE kit. And yeah, so this is something we have as an application for DynaSCOPE. But the take-home message today would be using our technology that we are a one-stop shop for all your sample to publication ready data needs. And our workflow can be instrument-free or automated, even for multi-omics, which I mentioned earlier. All our multi-omics options can be done with instrument or can be done manually. You don't need an instrument to work, you just need a pipette in that case. We can do up to 30,000 cells capture on one chip, and we have reduced cost per sample, which I will shortly mention in the coming slide too. But while we are at this slide, I can just talk about our kit. Like our pricing for our kits can also be discussed in our meeting one-on-one. So you can schedule a call with me, and we will be happy to help you with it. While we are in this meeting, I would like to talk about DynaSCOPE Single Cell Dynamic Library Kit, which was the point of interest for a lot of people around. And for this kit, we are actually offering a special limited time offer of 50% off for anybody who is interested from this webinar. We can actually offer it for $1,545 for 2 reactions. Normally, the pricing is about $3,090 for 2 reactions. This is the manual kit, and in case you're interested in Neo, we can also offer 4 reactions with the pricing increased accordingly. And you can have the instrument for free of cost for 3 weeks for trial, and you can have a look, and if it's a good use for your research, that's something you would like, or if there's any other multi-omics option that you would like to discuss, you can reach out to me on my email, which is included on this kit, on the slide, and Another thing I would like to mention is that our kits include everything to go from single cell suspension to library construction. So when I talk about the pricing for the kits here, it will include all the reagents, the chips, and the solutions that you need to go from single cell suspension to library construction. And yeah, that's all from my side right now. So thank you. If you have any questions, I'll be happy to take them now.
So the first question is there a pipeline for bioinformatic analysis for DynaSCOPE?
That's a good question. So we actually do have a pipeline. So our pipelines are all available on GitHub. So for all the multi-omics kits that we offer, including DynaSCOPE. So if you would like to look into the data, we have sample data available online, which you can refer to and you can download it for your user case.
Yes, yes. And another question about DynaSCOPE again. Is the metabolic reagent for labeling in DynaSCOPE added for similar hours for in vivo and in vitro?
Okay, yeah, so this depends. So it's not the same in vivo and in vitro, and even in vivo cases, depending on the tissue that you are targeting, it can vary. So, for example, for in vitro cases, if you're working with cell culture, it doesn't need to be as long of an incubation period as compared to inside some tissues. So if you're working with, for example, a tissue that's easier to access when once you inject it, it might not take as long. But if it's a tissue that's, for example, we had a customer who worked with embryo, and in that case, it took much longer. So it's a period of anywhere between 1.5 2 hours to 4 hours for the incubation period, depending on which tissue is of interest to you.
Yes, thank you, Harlan. A lot of questions are coming in. So one is, could you talk a bit about multiplexing, number of samples or cells that can be multiplexed?
Sure. So the multiplexing that we offer, it's a click-tag based chemistry, and we can do 16 samples in once. So you can have 16 samples in our DynaSCOPE with our scope chip. We have two versions of chips internally. So one can do 30,000 cells normally, but once you multiplex it, after that you can capture up to like 75,000 cells with it. So you can multiplex for because bioinformatically there's a pipeline to later then separate the cells. So it's possible to capture about 75,000 cells with the scope chip and 16 samples.
Yes. Can you please briefly re-explain how DynaSCOPE can be used in vivo?
Sure, I can. Yeah, so maybe I can go back to the slide.
Yeah.
I'm sorry for not going into detail here, but it depends on what we are looking at. So if you look at S4U, it's a label thiouridine, and you can take the label, it's included in the kit, and you can take the label and can inject it as an injection onto the mice. So it can be injected as an IP and intraperitoneal injection. And once it's injected, you wait for a couple of hours and then you sacrifice the mouse. So you can have a mouse, for example, you can have a control with no treatment, another for 10 hours of treatment, and another for 20 hours of treatment, for example. So you can have the three conditions and you can inject the label in all three of them and then look at the changes over a period of time. Yeah.
Thank you again about DynaSCOPE. Does DynaSCOPE target 30,000 cells per reaction as your other options?
Yes, it does. So we have DynaSCOPE also available as a standard kit, which can do 10,000 cells, but we also have a high density chip, which can do up to 30,000 cells with DynaSCOPE also. Yeah.
Yes. Can we get your kits sent from Europe rather than the US to Canada? Currently, sourcing few reagents from the US can be an administrative nightmare.
Oh, that's a good question. I actually might need to check about it. I honestly don't know the answer as of now. So I can understand it's difficult for Canadian universities, but I might need to check internally. Sorry about that. I will get back to you based on our internal answers.